Hello dear students, welcome again. As a part of pre-fertilization events in flowering plants, we had started discussing gametogenesis. In this gametogenesis, we had discussed the male gametes, how they are synthesizing from the microsporangium. We told that microsporangium has microspore mother cell, and this microspore mother cell is undergoing meiotic division to form microspore tetrad, and this microspore tetrad is maturing to form microspores, and these microspores are present within the anther. When the anther is dehiscing, when the anther is breaking open, these microspores are the pollen grains, these are rupturing out. And we also told that these pollen grains can be present in 2-cell stage or 3-cell stage. We said that if it is 2-cell stage, it is vegetative cell and generative cell. If it is a 3-cell stage, we said that the generative cell is undergoing mitotic division to form two male gametes, right? So we have seen the male gamete and microsporogenesis. In the megasporogenesis, the megaspore mother cell is undergoing meiotic division to form 4 megaspore cells, and these 4 megaspore cells, 3 of them are degenerating and only 1 megaspore is functional, and it is undergoing mitotic division, 3 consecutive mitotic division to form 8 nuclei 7-celled embryo sac. And we also have seen how it is formed in the female gametophyte. So this is in the megasporogenesis. So here we have seen both male and female gametes are synthesized, they are produced here. But how are these male and female gametes are coming together? Because these male gamete that is in the pollen grain, it is present outside, in the sense they are not in contact with each other, they are away from each other, and also they are non-motile. We also told that these flowering plants are producing the gametes which are non-motile, yet they are performing fertilization, and this is facilitated by an important process called as pollination. So now we are going to see the pollination, and to define the pollination, the transfer of pollen from the anther to stigma of a pistil of a flower is called as pollination. So this picture clearly represents, and you also have been studying the pollination. So these pollen grains which are present on the anther, they are carried from the one flower to other flower or in the same flower. So this is called as pollination. And pollination is carried out by the pollen agents. Some of the times plants, they require pollen agents. It can be insect, it can be water, it can be air, various other things. So based on the source of pollen, means based on the source of pollen, whether it is coming from the same plant or same flower, or it is coming from the different flower or different plant. So based on that, there are 3 types of pollination: Autogamy, Geitonogamy, and Xenogamy. These are the 3 types of pollination. In this picture, it is very clearly seen where in this there is a self-pollination, that is autogamy. In this autogamy, what happens is the anther of the same flower is landing on the stigma. The pollen grain of the anther is landing from the, so it is like the pollination is completed within the same flower. And the other ones, that is xenogamy. The xenogamy is cross-pollen, means the pollen grain from the anther of a different plant, it is landing on the flower of the different plant. So they are cross pollen. This is the pollination occurring between 2 different plants of the same species. Another one is Geitonogamy. So this is like self-pollination only, but this is occurring between 2 different flowers. So we are going to see them in depth. So starting with autogamy. Autogamy, we know that the transfer of pollen grains from the anther to stigma of the same flower. So when the when the pollination is completed within the same flower, we are calling it as a autogamy. For autogamy to occur in flowers, two conditions are required. So there are 2 minimum conditions required to have the successful autogamy. There should be a synchrony in the pollen release and stigma receptivity. When we say synchrony, what do you mean by synchrony? Synchrony means it is in the unison. When the pollen release and stigma receptivity, they should occur at the same time. Otherwise, if there is no coordination, then there will be no self-pollination. So there should be coordination, there should be synchrony in the pollen release and the stigma receptivity. Then only autogamy can occur. So this is the first requirement to occur self-pollination. The other requirement is the anther and stigma should lie close to each other. They should be remaining close to each other. They should be very close so that they are easily coming in contact with each other. Okay, so these are the two requirements for the autogamy to occur, and this autogamy is not seen in all the plants. Okay, and very important to remember this autogamy. Some of the plants, they are making the flowers which are only making the autogamy. Like some plants such as viola, oxalis, and commelina, they are producing two types of flowers. One type of flower is chasmogamous flowers. Another type of flowers is cleistogamous flowers. So these cleistogamous flowers are closed flowers, and whereas these chasmogamous flowers are open flowers. And these chasmogamous flowers, they are like normal flowers which are having the exposed anther and stigma. But whereas these cleistogamous flowers, they are like the bud. They do not open at all. So flowers which do not open at all are called as cleistogamous flowers. So this is very clearly evident that anther and stigma do not lie close to each other in these chasmogamous. It may be or it may not be necessarily. And anther and stigma lie close to each other in case of cleistogamous flowers. And in the chasmogamous, usually the cross-pollination or self-pollination can occur both. But here in the self-pollination is only pollination that is occurring in the cleistogamous flowers, because the anther and the stigma, they are lying close to each other, and this pollen grain cannot escape the flower. So it has to land on the stigma which is present in the same bud. So cross pollen, there is no chance of there is no chance of cross pollination in case of cleistogamous, whereas in the chasmogamous there is cross pollination chances because the flower is open and it is having the exposed anther and stigma. And the pollen agents are required for the chasmogamous flowers, whereas in the cleistogamous there are no pollen agents required. We are going to study what are the pollen agents and how are they useful in the pollination, and how flowers are adapted to have various pollen agents. So we are clear up to here how this chasmogamous is different from cleistogamous. So here flowers are adopting cleistogamous flowering. Cleistogamous flowering is adopted only for facilitating the self-pollination, and this kind of flowering is seen in viola, oxalis, and commelina. So here is the picture representing chasmogamous and cleistogamous flowers. So in the first picture you see it is chasmogamous flowers, and this is the example of viola, wherein the open flowers you can see here, this is like a normal flower. But the other one is cleistogamous flowers, which are, if you see here, they are like bud. They are unopened flowers. They are like bud, and this is carrying both anther and stigma inside. So the pollen cannot escape, and the pollen grain definitely lands on the stigma of the same bud, within the same flower. So here there is ensured pollination. There is ensured pollination here. So this is the advantage of cleistogamous flower. There is ensured pollination occurring even in absence of pollinator or pollinating agent. So even in the absence of honeybee or any other pollinating agent, there is ensured pollination occurring in case of cleistogamous flowers. Whereas in the chasmogamous, if there are no pollinating agents, there could be chance that they do not have pollination. So that's why, to ensure the pollination, they are having these cleistogamous flowers. Another greater advantage here is, and they produce assured seed set. Whatsoever, since the pollination is occurring, they are having definitely a set of seeds. So these set of seeds are required for carrying out life in the next generation. Means these seeds are shed out, and these seeds are again germinating to form the next generation plants. So that's how these seeds are formed definitely in case of these cleistogamous flowers. And this is example of viola flowers. Other kind of pollination is Geitonogamy. We told that here the pollen transfer is between the two different flowers of the same plant. So transfer of pollen grains from the anther to stigma of another flower of the same plant. So this is coming from the same plant but two different flowers. So it is a functionally a cross pollination. We can say it is a pollen which is landing on the different flower. It is a functionally it is a cross pollination, but it is genetically a self pollination. Genetically means since it is coming from the same plant, the genetics carried out in the pollination, the gametes which are carrying the genetics are the gametes that are carrying the nuclear material. It is of same type. So it is genetically it is a self-pollination, but functionally it is a cross-pollination. So it is a functionally cross-pollination because it involves the pollinating agent. There is a pollinating agent required. It can be water, it can be air, or it can be any insects, or it can be any other pollinating agent. But genetically it is autogamy, that is self-pollination, as the pollen grains come from the same plant. Since the pollen grain is coming from the same plant, this is a genetically it is a self-pollination or autogamy. So this is a Geitonogamy. The next kind of pollination is Xenogamy. In this Xenogamy, it is a cross pollination. So transfer of pollen grains from the anther to stigma of a flower from different plants of the same species. So here it is occurring between two different plants. The flowers which are coming from two different plants, the pollination is occurring between two different plants of the same species. So this is only type of pollination where the genetically different types of pollen grains are landing on the stigma. So here this pollen which is carrying the different type of genetics, different type of nuclear material present inside the gametes is carrying to the another plant. So what happens is there is a exchange of genetic material here between the two different flowers. So that's how this Xenogamy is a cross pollination, and these are the three types of pollination